see the unseen, change the unchanged. The innovation.
Why is the Omicron variant milder? Let me show you. Since 2019, COVID-19 has caused a worldwide pandemic. Recently, a new variant emerged in South Africa and was designated as the fifth variant of concern, named as Omicron. As a highly infectious Omicron variant, it also rages in parts of China. At the beginning of March 2022, Shanghai are coping amid harsh COVID-19 lockdown. The Omicron variant is the most mutated variant, and its high transmissibility and immune evasion ability have raised global concerns for antiviral therapy. In addition, multiple recent studies have shown that the Omicron variant exhibits attenuated replication capacity and produces substantially attenuated lung pathology, indicating that the pathogenic ability of Omicron variant is milder. The available studies suggested that whether vaccination is not correlated with the reduced pathogenicity of Omicron. This fast-moving variant poses new puzzles. Four major problems scientists are racing to answer. How is it so transmissible? Is it less severe? If so, why? What is the complete immune response to Omicron? What comes next? Mutation studies in the spike protein RBD domain have provided a plausible explanation for altered transmissibility and antibody resistance. However, the second question, the molecular basis for the attenuated pathogenicity and replication capacity, remains elusive.
The high-frequency spontaneous mutations of Omicron spike protein result in stronger transmissibility and immune escape. So, whether there are spontaneous mutations in other structural proteins of SARS-CoV-2. If so, what is the physiological relevance? Our previous work found the envelope protein 2E forms a homopentameric cation channel. 2E channel alone is sufficient to induce cell death, provoke cytokine storm, and even cause acute respiratory distress syndrome-like damages in vivo. The channel inhibitor exhibits excellent antiviral activity in vivo. Thus, 2E channel may represent a new class of dual-function targets against SARS-CoV-2. Here, we focus on the mutations of 2E channel. Based on sequence alignment, a mutation T9I caught our attention. T9I shows a much higher frequency than the rest mutations. In addition, the allele frequency of T9I in all statistical samples is higher than 99%. Whether T9I still retains channel activity was first investigated. We used the planar lipid biliary workstation for electrophysiologic experiments. Let me show the workflow. First, look for the field. Apply phospholipase to the small holes. After the planar lipid biliary formed, the baseline could be recorded. Subsequently, the protein added. When the channel assembles, the classical single-channel currents could be recorded. We observed T9I was able to form ion channel also as wild type. However, compared to wild type channel, there are two significant changes in T9I properties. The ion selectivity changed, and the proton sensitivity reduced. As we know, SARS-CoV-2 traffics to the lysosomes for egress by lysosome deacidification. First, we found both wild type and the mutant channel are co-localized with the lysosome. Through the flow cytometry analysis, the capability of deacidification of 2E channels in lysosomes was significantly impeded by the mutation. Worth to note, pre-expression of mutant significantly attenuated viral loadings compared with wild type. In the end, we tested the virulence of T9I mutant. As expected, the killing cells capability of T9I was dropped around 150 times as well as the cytokines production. Overall, we report for the first time that a high-frequency 2E mutation T9I of Omicron forms a non-selective ion channel with reduced acid sensitivity compared to wild-type channels. In addition, the mutation channel caused less cell death and a weaker cytokine production. The channel property changes might be responsible for the Omicron variant releases less efficiently and induces a comparatively lower level of cell damage in the infected cells.
As Omicron infections are progressing, there are chances that virus accumulates new mutations. The notion that viruses will evolve to be less virulent to spare their hosts is one of the most persistent myths surrounding pathogen evolution. Unlike viral immune escape and transmissibility, which are under strong evolutionary pressure, virulence is typically a byproduct. Viruses evolve to maximize their transmissibility, and sometimes this may correlate with changed virulence. Those mutations introducing maximal transmissibility and enhanced virulence may kill the host and suppress the transmission. In contrast, those mutations attenuating the virulence could be retained in the variants with maximal transmissibility. Before the emergence of Omicron variants, the mutation T9I has appeared in the E proteins of other SARS coronavirus 2 variants, including Alpha, Beta, Gamma, and Delta. The prevalence frequency of T9I in these variants was less than 0.2%. We speculated that the mutation T9I may originate from these previous variants and was retained in the evolutionary screening, perhaps due to its reduced virulence. Notably, in the latest emerged Omicron XE strain, T9I was still retained and is the only mutation identified in its envelope protein. However, we cannot exclude the possibility that the origin of T9I is just a coincidence as the milder severity with Omicron. Our study provides valuable insights into key features of the Omicron variant and critical information for pathogenicity evolution. Moreover, it also reminds scientists that in addition to spike protein, mutations in other SARS coronavirus 2 proteins also deserve attention. Thanks to all professors and fundings. That is all. Thank you.
Pursue excellence.